It's important for us to understand the patterns that we observe today and how they were formed so that you can have a good prediction. So you first need to understand what is behind this pattern that we see today. So this is the map of fish diversity, so ranging from few species, maybe 50 species overlapping, until many species, like 3,000 species overlapping. And so the important questions to be asked here is what processes leads to this pattern. So you clearly see that there is not, it's not homogeneous, there is a gradient and so on. So there are many ways to ask this question. So one of these questions would be, why do some places have a higher diversity than other places? And is it true? Yeah, I think it's quite long. Better like this? Oh, should I start from the beginning again, or no? Okay. So yeah, and why aren't some species everywhere? So you see clearly that Napoleon has no idea, but the clownfish here is giving us some clues. So maybe we should look at how species are formed and how they are extinct. And today we're going to focus into how the species are formed. So what I'm going to explain here now, it's very basic because we heard that there will be first year master or bachelor students. So forgive me if it's very redundant, but if it's not, pay attention because this will help you throughout the other presentations. So we know that individuals of the same species, they are able of reproducing, right? And this reproduction will lead to an exchange of genetical material. And this is what we call a gene flow. And an interesting question is, okay, what happens if this gene flow is stopped? So to explore this in a more easy way, I will start with elephants, and then we'll go to fishes. And one way of understanding speciation would be the allopatric speciation. So in this example, you can imagine like a continent, this squared green continent full with white elephants, and they're all the same species. And as I said, they are same species, there is a lot of gene flow among them, they're keeping their population homogeneous. But what happens if a barrier appears? So let's say a river, and these elephants, they're really afraid of water, they don't like swimming. So consequently, you have an interruption of gene flow between these two populations, or we call them subpopulations. However, you still have some gene flow within these subpopulations. And what happens is, over millions of years, you accumulate mutations, natural selection will act, and at the end, you have two different species. And they will continue different, even if you bring them together, if you take this barrier away. So, this is all the idea behind this allopatric speciation, and allo, other, and patric, fatherland, so other place of species that are separated. So, it's easy to imagine this barrier for terrestrial species, right? But then we tend to think fishes can go everywhere. It's very hard to understand this barrier for fishes and marine species, but they also suffer gene flow, or these interruptions in gene flow. And it's all related with suitable sites. So the question is, these barriers, or these interruptions of gene flow, they're really important, they're really related to suitable sites. And these suitable sites are formed by geological and climatological processes. So I'm going to explain a little bit of these suitable sites using plate tectonics. So it's very easy for us to imagine that the Earth is just stable as it is, but we know that it's formed by many plates. These are the tectonic plates, and they're dynamic. So imagine here you have a very simplified cut of the Earth. And then on the blue side, I didn't imagine it would be so high and big, but imagine, so the blue is the sea, you have the plates, tectonic plates, and they are forming actually by the underwater topography. You can see a suitable site for clownfishes. So it's not too deep, it's not too shallow, and you have there a suitable site. But with time, these plate tectonics will move, so they're colliding. And what happens is that you have a split of suitable sites. So now, in the middle, now it's too shallow, or it's not water, and you have two different sites. And this consequently leads to an interruption of gene. And as you can see, this is a real example. So you can kind of follow the separation of the suitable sites by this green species, which was separated around 30 million years ago. And then by the formation of the Panama Channel, you have again a split. And well, all this this continental drift idea, as I said, is like not tectonic but also climatic. So in order to construct our models, we use climate data and geological data. And here you can see a map. So the white area are land surface, and then you see the water, and you can see a band. So the band, the colorful band is showing the tropical area, and the white blue is shallow water, so suitable sites for coral reefs. And you can see how they change over time. So from 138 million years ago towards present. And what we do, basically, we model. So here's an example of the modeling of one speciation event. So you have a red species occupying a site, and then this site, which is occupied by one species, is splitted, so it's separated. And when it's separated over longer than 1 million years, we assume that we have two different species. And now you see the blue one and the green one. And even though if they come together later, they're just two different species. And here we are showing basically the hotspots and the diversity through time, so 40 million years, 50 million years, and 1 million years ago. And on the left-hand side, you see what we know by fossil records, and on the right, it's a match with the models we are using. So you can see that we can match quite well the development of this biodiversity and how this tracking back this biodiversity. And especially if you look at the TTC, for example, the high diversity and so on. So, yes. Now, but anyways, you can look, as Luke already said, you can look at this diversity in different ways also, and Julia's going to talk more about this now.